Another topic I would like to cover today is the nanopore sequenator. What makes it different from previous generations of sequenators in the recognition of the DNA chain of nucleotides? It is the number of pairs it can make at the same time. If the old samples made only about 300, that is, when we studied the human genome, we studied the longest sections of 300 nucleotides, now it is up to 2 million. The chain can make up genes that we study at the same time. That is, in fact, we can very quickly study viruses, various pathogenic samples, and, of course, they will be very much in demand in criminology, in agriculture. In general, biotech is such a through project. It unites the efforts of different industries. That is, industry, the agricultural complex, pharmaceuticals, medicine, and, by the way, processing in environmental issues is also in active demand.
Exactly 2 years ago at the forum here, you supported the concept of creating new bionic prostheses and gave us an assignment to try to make the world's first sensory prostheses. Last year, we created the Center for Cybermedicine and Neuroprosthetics at the Center for Brain and Neurotechnology at the FNVA. And today, we are showing our products. We have developed in two directions. We created the first bionic prosthesis of the hand with a sensor due to the fact that special pressure sensors were inserted into the fingertips of this prosthesis. In order to transform information into electrical signals, a device was developed. The device is a converter of electrical impulses and forms stimuli. These stimuli must be transmitted to the peripheral nerves, the preserved peripheral nerves of the hand, in order to pass through the back of the brain, the opposite hemisphere of the brain, and become conscious as a sensation. Therefore, a special neurochirurgical electrode was developed for implantation into the peripheral nerves, with a special, unique technology for neurotransmission of this nerve. And in fact, we have achieved that. A person perceives the soft and hard form of the object with the prosthesis, which takes the weight of the object, and so on.
We begin to print without any threat of an immune response from the body. These are such universal, generally acceptable cells. Here is printed on this copy, of course, an enlarged valve of the aorta of the human heart, one of the most susceptible diseases of our organs. And accordingly, we move forward to a more complex system. We can print blood vessels, functional tissues, muscle tissues, but the heart is a unique creation of nature where electrical impulses are born. And such a complex mechanism of printing is not yet possible. We decided to use our radioisotope sources, nuclear batteries. Thank you very much. You gave a premium to young scientists, including for this invention this year. We offer them for active use in cardiostimulators. And I hope sooner or later we will be able to present such a full-fledged implant, biological engineering heart, to you in the month of Famba.